In today's lesson, we're going to be looking at DNA replication, and our learning question is, how does DNA actually copy itself? These are the keywords associated with today's lesson, and you can find these in your textbook. Please make sure that you do the relevant reading. This is our learning objectives associated with today's lesson. We're going to be focusing in mainly on how DNA replication occurs and how it uses these enzymes to do that. Now, the structure of DNA, we've looked at this diagram before. You can see that it goes in two directions, and that is described as anti-parallel. So they're parallel to each other, but what you have is one strand flowing in one direction and the other strand flowing in the other. So you can see that using those red arrows there. Now, these little numbers help us to orientate the molecules, and when we look at these numbers, and you can see that it's 5 and then a little mark there, and what this denotes is the term 5', and DNA will always run from 5' to 3', and you can see that in the other direction, 5' to 3'. Now, when we're looking at them both the same way, we can see that one is anti-parallel to the other. Now, the way that DNA is orientated itself is because of the molecular structure, and there's a lot of biochemistry behind that. So all we need to know is that DNA runs 5' to 3', and that one strand is the opposite of the other, and this makes them anti-parallel. Therefore, one strand is called the leading strand, and the other one is called the lagging strand. Now, when we look at the replication of DNA, it's really important that DNA copies itself exactly. We know that if there are mistakes in copying of this code, it can lead to mutations, so that could be a tumor or a cancer, or it could mean that a protein isn't coded for correctly and it malfunctions. Now, we know that with mitosis, which is cell division, everything is copied exactly. And what this does, it means that every cell has the exact same genetic code in it. Now, an example here that I'm showing you is a zygote, which is a fertilized egg cell. When that first undergoes its rapid cell division, every single one of these smaller cells are identical to the original zygote. At a later stage, these individual cells may differentiate to become different organs, but at the start, it's vitally important that they all have the same genetic information. So now we're going to be looking at replication of DNA. It occurs inside the nucleus of eukaryotic cells. Remember, we have membrane-bound organelles, hence we are eukaryotic. And we need to think about what we need to make a brand new strand of DNA. So the first thing we need is a template. So if you're going to copy down these notes, you're using these slides as a template to copy down the individual letters to make up the sentences that will make sense. DNA requires a primer, and this is a short piece of code that tells DNA where to start copying from. So you can think about it as a bookmark. So we've got a template and we've got our primer. And then we have a series of enzymes. Now you'll remember that enzymes are protein-based catalysts, and remember these speed up the rate of reaction without being changed themselves. So they're vitally important for life. The first enzyme we're going to be looking at is DNA helicase. So DNA helicase will unwind that DNA double helix, so you can remember it that way, and it unwinds that twisted structure into the two parallel strands. Then we have DNA polymerase. Now remember, the word poly means many, so we're adding many nucleotides together. And then finally we have DNA ligase, which is a gluing enzyme which binds DNA fragments together. Now, in addition to our template and our primer and our enzymes, we also need lots of free nucleotides to make that DNA strand. So you need to build that DNA strand using nucleotides. And lastly, because we are forging new chemical bonds, we must have a source of ATP to ensure that we can bond those free nucleotides together and break bonds when we are unwinding that DNA double helix. So this little animation just shows you what's happening here. So take the time to watch it a few times. First of all, we have the enzyme helicase, which is unwinding and unzipping that DNA molecule. So instead of it being twisted, we have two parallel strands. We saw the primer coming in that was telling us where to start coding from. I'll come back in a second. And then we have the DNA polymerase, which will add the nucleotide fragments. So here we are, we have the primers, which is telling us where to start. DNA polymerase will start construction. It will add those nucleotides from that primer. And you can see here the leading strand goes in the 5' to 3' direction, whereas something different happens on the lagging strand. We get these short fragments that occur because of the anti-parallel nature of DNA. And this gives us fragments. So we're going to look at each of these cartoon images in turn and describe what's happening. So the first stage we could see here was DNA helicase caused that DNA double helix to unwind. So it's no longer twisted, it's parallel. And then we have unzipping. Now, unzipping is when those weak hydrogen bonds between the nucleotide bases start to break. Now, the reason why we need to have unzipping is because we want to expose that DNA code. So the individual bases, the adenine, thymine, cytosine, and guanine, the arrangement of them along the DNA strand makes a code. So we can't actually see what's been written there without looking at that code. Next thing that happens is a primer attaches to the DNA. So remember, a primer is just a short section of DNA that helps the DNA polymerase enzyme know where to start building from. Okay, so once we have got our primers in place and DNA polymerase is there, free nucleotides will start to pair up with the exposed DNA bases. So this black line here, we can't see it because it's just a cartoon, but you'll have DNA bases that are free, and you will have free nucleotides that will attach in a complementary way. Now, the enzyme DNA polymerase helps the nucleotides to bond together. Now, in this diagram here, you can see this is our template strand along the top, and these are the free nucleotides along the bottom, and you can see the weak hydrogen bonds forming between complementary base pairs. On the backbone, between the phosphate and the sugar, we have a covalent bond. Previously, I've called it a sugar-phosphate bond to help you know where it is, but you should try and use the term covalent bond, and this forms the backbone of DNA. So you can see in this cartoon down here, you have got the template strand of DNA, and this blue region is just showing that it's a copy. So we have got complementary base pairs here and here, and you can see them here. And what is happening is the DNA polymerase enzyme is helping these nucleotides to bind and form bonds, and as that moves along, the strand of DNA that's been copied is being elongated. Now, in the animation, we looked at we noticed that you have this nice smooth line going from 5' to 3' in the copy direction. So this is the leading strand. On the other strand, the lagging strand, the information was being copied, but it was being copied backwards. Now, this is copying from 3' to 5'. Now, it can't copy in that direction easily. So we end up with these short fragments. Now, these short fragments are actually known as Okazaki fragments because of the scientists that discovered them. But all you need to remember is that the leading strand copies continuously from 5' to 3', whereas in the lagging strand, the DNA is copied backwards and we have these small fragments. Now, in order to make a continuous strand of DNA, the fragments have to be glued together by the enzyme DNA ligase. So again, in this cartoon picture down here, we have our seamless leading strand copying from 5' to 3', and we have our lagging strand copying in small fragments from 3 to 5. Now that our DNA has been copied, the fragments are bonded together by the enzyme DNA ligase. DNA helicase will actually rewind the DNA molecules because you don't want DNA to exist completely spread out. After that's happened, we have two identical strands of DNA that have been formed. Now this diagram, even though it looks a bit busy, it really does a good job of summarizing it. So if we look at our chromosome, and when it's unwound, we can see at the most basic unit, we have this wound up double helix of DNA. Enzyme DNA helicase unwinds and unzips this double helix, so we can see that these bases are exposed. A primer comes in and tells DNA polymerase where to start copying from. So we can see here that these little yellow blobs are the polymerase, and it is adding free nucleotides in a complementary fashion, and it will keep doing that until the full molecule is coded for. Now, DNA replication is described as semi-conservative. The word semi means half, and to conserve is to keep. So the reason why it's called semi-conservative replication is because half of the original DNA is kept in the new double helix. So we have two new strands that are formed from DNA, but one is the original strand, and you can see that here in this diagram. So this is our unwound DNA, and this is unzipped. So then we have these free nucleotides that come and bind to either side of that DNA. So when we have two new strands formed, they are semi-conservative, the red one being the old and the blue being the new. All right, so now let's do a summary of DNA replication. Let's look at this diagram here. What we can see is that the enzyme DNA helicase will unwind and unzip that DNA molecule. And what we have here is DNA helicase doing that, and you can see that this is now parallel strands rather than all wound up, and the DNA bases are separate from each other, so that code can be read. Second thing that happens is that we have short primers, that's these little sections here, which will attach to the start of the DNA molecule for where we need to start reading from. And as a result of those primers being bonded to, DNA polymerase knows exactly where to start adding nucleotides. So this is DNA polymerase here, and you can see them moving in the 5' to 3' direction at the top and the 3' to 5 in the bottom. Now, as that DNA polymerase molecule moves along the strand, we start to get elongation of that template DNA being copied in the leading strand from 5' to 3'. The lagging strand, on the other hand, has to have a series of primers because we can't seamlessly add nucleotides. So we end up with these short fragments of DNA known as Okazaki fragments. To ensure that DNA is seamless, these fragments are glued together using the enzyme DNA ligase. Now, after DNA ligase has glued the fragments together, the enzyme DNA helicase comes back in and you have the rewinding and rezipping of that DNA. And we have the two brand new fragments that are semi-conservative. Now, this is a really good video to watch by the Amoeba sisters that summarizes in a really nice cartoon way the process that we have described. I'm not going to watch it just now, but I do encourage you to do so. For today's lesson, what I would like you to do is to complete page 6 of your booklet and read or reread page 235 of your textbook and answer the questions on page 7 and 8 of your booklet. So you should be completed up to and including page 8 of your booklet by the end of this lesson.